Hello, in this video we will discuss about the cellular stress. So what is the stress? Stress means the damaging of something due to external or internal causes of the human body. So in an organ, here I am drawing the in an organ there is a balance between the growth and death of the cell that make it up, which is called tissue homeostasis. So the tissue homeostasis is very important for the normal life. If the cell death will increase, so when cell death will be increased, so the cell division will be decreased, lead to cause aging. So remember that things aging is due to the increase of the apoptosis. Here I am drawing the person which contain a different type of cell, and cells contain tissue, tissue contain organs. So here I am drawing the tissue, which that is, for example, I am just drawing the single cell for understanding the basic concept. So first of all, we should need to understand the heat shock response. So what is this? Heat shock response mean when heat will increase near the cell will lead to increase the heat shock factor, and this heat shock factor is a signaling molecule, and heat shock factor will bind with the DNA in the nucleus, and heat shock factor is different type. Here I am just understanding the general concept. So the stress also physiological states, pathophysiological states, and small molecule modulators. There are many toxins etc also. So in this way the heat shock response is due to the heat shock factor. This heat shock factor is the heat shock factor will bind with the DNA. After the binding with the DNA will lead to heat shock enzymes and heat shock protein, and this heat shock enzyme protein will lead to heat shock protein, for example, is lead to cytoprotection, and misfolding of the protein will inhibit, and the signaling pathway will increase, and apoptosis will lead to decrease, and aging will decrease by this pathway. So how it's possible to decrease the aging? Let's begin to understand, because aging is very crucial to lead apoptosis. So that is, here is the heat and cold. This is the heat stress and cold stress. Two type of stress can be mainly more heat, for example the 50 degrees Celsius, and more cold weather, for example the minus 4 degrees Celsius, which that affect the cell. For example, here's the phospholipids cell membrane, which that will lead to trigger the PIPK kinase, and this in this way the PA which that is will trigger to IP3 pathway, and this IP3 pathway will lead to increase the calcium also. So the calcium will come, efflux of the calcium and influx of the calcium, as well as endoplasmic reticulum is also produced due to the IP3 sensitive calcium gated channel. And in this way the calcium gating channel will and binding protein and cytoskeleton changes due to the calcium, and the calcium is also important for the cytoskeleton maintenance. And on the other hand, the stress produce the reactive oxygen species. So in this way all things, the protein and cytoskeleton changes, which that will lead to increase the NADPH oxidase enzyme. This NADPH oxidase enzyme will lead to trigger the reactive oxygen species. This reactive oxygen species will be produced, and in this way here you can see the MAP kinase cascade, as well as the CDPK kinase, which that it will lead to bind with the DNA as a transcription factor to produce that protein, which that is synthesized during the translation to tolerance for the cell. So this is the heat response, and in this way the cell will homeostatic formation and not apoptosis. While in the case of cold weather, so the calcium, for example, here is the calcium, which that is influx of the calcium into the cytoskeleton changes due to the calcium bind with the troponin and centriole for the changing of the calcium, and in this way the CDPA and CRT, which that is the transcriptional factor, and this will lead to transcriptional factor by different pathway, And here is the rigidity of membrane due to the cold stress will lead to also increase the diacylglycerol kinase cascade, and in this way the trigger the DAG kinase pathway, diacylglycerol pathway, which that means the diacylglycerol are present in the plasma membrane. And here you can see the cytoskeleton and ABA and NO, which that is also uh, basically is produced from the cold stress. So the here is you can see the CDPA and CRT, which that is the transcriptional factor, will lead to increase the CRT, and in this way you can see the accumulation of stress response will be occurring this way. And here is the ABA will trigger the MAP kinase pathway, will cascade of the MAP kinase to convert MAP kinase and MAP kinase. And this MAP kinase will bind with the DNA where the transcription factor and specific genes are present. And in this way the MAP kinase will trigger to increase that protein which that is desirable during the cold and stress. And also important thing you should need to know that the UCP1 is also produced for the mitochondrial heat production through a brown fat. So in this way here you can see this the DAC kinase phospholipase C pathway, which that is the enzyme used to phosphatidic acid production, and the reactive oxygen species will produce. And this reactive oxygen species will lead to damage the DNA, but in this case accumulation of stress response genes antioxidant will be produced and reactive oxygen species. So the antioxidant will lead to decrease the reactive oxygen species and stress tolerance, while the protein synthesis for resist the stress and adaptation in harsh condition is very important. And in this way that protein which that is very desirable for during the cold stress, and the food which that is we are taking as an antioxidant is also play important role for the stress response and maintain and homeostasis for the cell. So the starvation is the different stress. In this way the glucose decrease and glycogenolysis and gluconeogenesis will be occurring, and energy stress. So this is the energy stress because we are not taking too much energy, not taking too much food. So in this way the glucose will be decreased, and during the glucose decrease the hypoglycemia will be occurring in the blood. After the hypoglycemia the blood will not glucose allowed to move into the cell because the insulin level will be decreased and amino acid will be increased, so the proteolysis will be increased, and as sufficient of amino acid and energy stress during the starvation possibly will lead to increase the mitochondrial dysfunction, because the mitochondrial dysfunction is due to the decrease of the ATP and on the other hand the decrease of the glucose, and there are a lot of things which that is I am not mentioning here, but next video we will discuss also. So here is the growth factors. These growth factors basically bind for the proliferation and cell differentiation, also mitotic division. There are a lot of things which that is related to the cell growth. So here is the PI3K3 pathway after the phosphorylation of the phosphoinositide 3 kinase protein 3, and in this way this is the growth factor will lead to tyrosine kinase pathway, tyrosine kinase receptor. So here is you can see this is the ATP will decrease due to the mitochondrial dysfunction, as well as the starvation will lead to trigger the mitochondrial dysfunction and for apoptosis. So the cell apoptosis is very important for maintaining the energy, because a lot of cell will require more energy but not more energy, so the apoptosis should be, because the apoptosis is very important for decrease the energy supply, and that apoptotic content will convert into the energy for further vital function. So here the PI3K will trigger the AKT to the phosphorylation AKT. So the here is the You can see this is the AKT will trigger the AKT. You can see this is the AKT activated AKT. Sorry, this is this diagram is, you can understand this is the reverse. So the AKT activated will increase the mTOR pathway with the mTOR complex one with raptor protein. So the mTOR pathway inhibition means the inhibition of growth of the cell and promote the cell apoptosis. So what that means? That means you should need to understand the inhibition of the growth due to the AMPK pathway, which that is after the decreasing of the ATP will lead to AMPK activation to AMPK activation, and that will lead to inhibit the mTOR pathway, and that's okay. And in this way here is the export reactive oxygen species. So reactive oxygen species will export. And here is the phago4. So this is the phago4. This phago4 will decrease basically to become a autophagosome. So this autophagosome you can see and understand that is the autophagosome formation due to the inhibition of mTOR pathway, while the mitochondrial dysfunction will lead to increase the phago4 and the AMPK pathway will also, and this autophagosome will due to the decrease of the NAD plus and ATP decrease as well as the perylation will increase and the phagosome will be increased, and in this way the basically the perylation means the damaged DNA recovery. So the damaged DNA recovery is very important, and that protein will produce from that apoptotic cell for changing for recovery of the DNA of another cells, which that is which that is sustained. So here you can see this is the reactive oxygen species also export and damage the DNA. After the response of the damaged DNA will lead to increase the PARP, which that is the PARylation will be occur, and this PARP gene will be activated after due to the decrease of the NAD plus and decrease of the ATP. And this is PARP mean the poly ADP ribose polymerase enzyme issues, and the DNA repair will be occurring. In this way the DNA will be repaired. So I hope you are getting this concept, and this is the cellular stress, and this is the short video. I hope you like this video. If you like this, so subscribe.